The stomach. The stomach is a holding sac in the superior left quadrant of the abdomen, where the mechanical and chemical digestion continues onto the food products or the bolus. Under normal conditions, this can house between 3 to 4 liters of food, drinks, and saliva that enter into the stomach daily. And they spend about 2 to 6 hours here processing. Food is then mixed with the secretions from the stomach and then released from the stomach into the small intestines in a fluid called chyme. The stomach initiates the chemical digestion and breakdown of proteins and fats. Alcohol and aspirin and some drugs are absorbed directly inside the stomach. Here is an overview of the anatomy of the stomach. The stomach is a J-shaped organ that has a greater curvature of the stomach, which is going to be the larger convex surface. It also has a lesser curvature that is a smaller convex surface. The cardia is the first 4 regions of the stomach, which is a narrow superior entryway into the stomach from the esophagus. The cardiac orifice is a terminal opening where the cardia meets the esophagus. So that will be right here. And then the fundus is a dome-shaped region superior lateral to the cardia, and it has a weaker muscular contraction and contains a slightly higher pH in its lumen. The body of the stomach is going to be the largest region and is inferior to the fundus. The pylorus is going to be a narrow, medially directed pouch that forms the terminal region of the stomach. And then we also have the pyloric sphincter that controls the entry of material into the duodenum or the small intestine. The ruga are these folds that increase the surface area inside the stomach. The gastric pits are a unique set of histology found inside the openings for secretion into the stomach. Here is a cartoon image showing you that. The gastric secretions made by the gastric pits include secretory cells in the stomach that produce about 3 liters of gastric juices per day. So over here you can see within the pits there's parietal cells, chief cells, G cells for gastrin, D cells for somatostatin, and then at the base we have the interchromatin cells. We will talk about each of these cells in the next slide. The chief cells are going to be the zymogenic cells or the peptic cells. These are the most numerous secretory cells that secrete packets of zymogen granules, primarily containing a compound called pepsinogen and gastric lipase. Pepsinogen gets activated into pepsin with the help of the hydrochloric acid. It also contains a hormone receptor that stimulates the pepsin release into the stomach. So the key functions are that the pepsinogen is activated to pepsin, and this helps break down and denature proteins into oligopeptides, and the gastric lipase begins the breakdown of the lipids and fats. The next set of cells we'll talk about are called the parietal cells. Parietal cells, also known as oxynitic cells, are responsible for the addition of the intrinsic factor and the HCl hydrochloric acid into the lumen. Intrinsic factor is going to be the compound protein that is required for the absorption of vitamin B12 in the small intestine. HCl stands for hydrochloric acid, which is going to facilitate in chemical digestion, protein denaturization, pepsin activation, and enhances enzymatic activity and kills most of the microorganisms that enter the stomach. It also secretes a little bit of bicarbonate to help begin to neutralize some of the acid contents later on. Next, we'll talk about the G cells. G cells are an enteroendocrine cell that secretes the hormone gastrin into the blood that stimulates the stomach secretion and the motility. It also has receptors on the basolateral side, parietal cells, that also help stimulate HCl release. Because of this, it also helps stimulate pepsin from the chief cells and histamine from the ECL interchromatin cells. So the fourth cell is the enteroendocrine cells, ECL cells. These cells are located inside the gastric mucosa and are responsible for producing and releasing histamine. Histamine will then in turn bind to the H2 receptors on the parietal cells that will then stimulate more HCl and hydrochloric acid secretion. When these ECL cells are stimulated, they release histamine into the surrounding tissue, and it diffuses to the parietal cells and triggers that response. This cascades into the activation of the proton pump. The activation of the proton pump results in the secretion of protons from the parietal cells into the lumen. And then the last set of cells are called D cells, which secrete somatostatin. These delta cells are specialized inside the pancreatic islets of Langerhans as well as inside the stomach. Their main function is to release somatostatin. It also regulates glucose metabolism. Its main function is to trigger the release of pancreatic enzymes and also inhibit hormone secretions. Somatostatin is released by D cells and it has inhibitory effects on the release of other hormones like somatotropin or growth hormone. It also suppresses the thyroid-stimulating hormone, insulin-like growth factor, and as a result, it will inhibit gastrin and histamine. So it turns off the process of breakdown of contents in your stomach. The parietal cells use active transport to secrete HCl into the stomach lumen. The proton pump is an enzyme called the proton potassium ATPase located on the surface of the parietal cells inside the stomach lining. It plays a critical role in the production of HCl. When activated, it binds to histamine on the H2 receptors, and it triggers a signaling cascade that leads to the activation of the proton pump. ATP gets converted into cyclic AMP, and as a secondary messenger, acid production begins to start. The transported protons moving out are exchanged for potassium with the help of ATP, and then this proton right here gets combined with chloride inside the lumen of the stomach to create HCl. The histamine will trigger the activation of the G protein stimulatory pathway that will trigger more cyclic AMP. PGE2, prostaglandin E2, is going to inhibit this pathway, which will block the production of acid. Acetylcholine and gastrin are both going to increase calcium, which also will turn on the protein kinases to trigger activity of the ATPase for proton potassium. Therefore, histamine, acetylcholine, and gastrin all favor the production of acid, whereas PGE2 decreases the production of acid. A focus on disease. Peptic ulcers are chronic, solitary erosion of the lining of the stomach or the duodenum. Gastric ulcers are peptic ulcers that can occur inside the stomach. Duodenal ulcers are peptic ulcers that occur in the duodenum. Symptoms of an ulcer include burning pain in the epigastric region, nausea, vomiting, extreme belching, and tar-like stools. In worst-case scenario, perforation of the organ may occur left untreated. Helicobacter pylori is a bacterium present in 70% of the gastric ulcers and over 90% of duodenal ulcers, which will eventually erode the stomach lining. Some of the key treatment options include using antacids, antibiotics, dietary changes, and also PPIs or proton pump inhibitors. Anything to reduce acetylcholine, gastrin, and histamine are going to be good. Stomach or gastric motility. The smooth muscle activity in the stomach has two primary functions: mixing the bolus with the gastric juices to form chyme, and emptying the chyme from the stomach into the small intestine. Gastric mixing is a form of mechanical digestion that changes the bolus into the chyme. So gastric emptying is the movement of chyme from the stomach to the duodenum. It starts with a wave-like peristaltic contraction that moves the chyme through the pylorus towards the pyloric sphincter that produces a pressure gradient that pushes small amounts of the chyme through that little sphincter. As it squeezes and squeezes, the peristaltic wave ends, pyloric sphincter closes and pushes the stomach content back towards the body of the stomach. Retropulsion is the reverse flow of the chyme back into the stomach body as pyloric sphincter closes. This allows more mixing to occur. So as food mixes with the stomach secretion to form chyme, the food enters the stomach. There's going to be this tonic smooth muscle contraction. This is stimulated by a set of cells called the interstitial cells of Cajole, which are a unique set of cells, or also known as pacemakers, for moving the food. These pacemakers are located in the longitudinal layer of the smooth muscle, and they have spontaneous depolarization of about 4 times per minute. This then leads to the phasic smooth muscle contraction, followed by the upper stomach expansion, keeping the constant pressure, and continuously squeezing. The mixing of the waves push the chyme towards the pyloric sphincter. As a result, the stomach distends, and now more gastrin gets released to help with the breakdown of the food. Eventually, when the pyloric sphincter relaxes, the food gets pushed into the duodenum, and now retropulsion occurs and it repeats itself over and over. Sensory signals are sent to the brain, specifically the medulla oblongata, and result in motor responses to increase motility and secretion of the gastric cells. If there's a presence of food, particularly protein in your food, this causes the release of gastrin and increases the force of contraction. And it causes the closing of the pyloric sphincter. Protein actually helps encourage the activity of the stomach. In the final phases, other hormones like cholecystokinin, or also known as CCK, is released in response to some of the fatty chyme that causes a decrease in stomach motility and helps inhibit the release of gastrin. Secretin is another hormone that is released in response to acidic chyme that causes a decrease in stomach motility and a decrease in secretory activity. both of these hormones are going to be talked about later, which reduces the properties of the stomach, which reduces the properties of stomach activity to help neutralize the acid.